It has been a pleasure. Never been to Bulgaria. It has been a great time. So almost everyone in this session apologized because they're not working on genomics. I'm definitely not working on genomics. However, there are a few topics that you're going to see, or themes that you're going to see in my talk that you have seen in others. So there is in this disposition of KRAS4B on lipid bilayers, you're going to see that KRAS4B has a globular domain. It's actually localized into the membrane by a disordered domain, and then the properties of that disordered domain actually determines largely the behavior of the protein on the lipid bilayer. So why do we care about KRAS4B? KRAS is sort of at the initiation, close to the initiation sites of many different pathways that are relevant for cell signaling, and it's a switch protein. So it's a small GTPase, actually goes from inactive state when it's in the GDP-bound form into an active state when GTP actually binds to it. That induces a conformational change, redigitization of a certain loop that induces or promotes or facilitates the binding of most of these proteins that you see here. Many of these proteins are called effectors, and then these proteins play roles in recruiting other proteins. Some of them are kinases that actually start a cascade of a signaling cascade. So why do we care about KRAS? If you look at the pathogenesis, there are many different cancers that are related to KRAS, in particular, or to RAS. KRAS is one of the most worst players into cancer, and more than 30% of the cancers are driven by some KRAS mutation. Of particular importance, pancreatic is 95% related to KRAS, and it's actually related strongly to one mutation in the loop that in one of two very significant loops of this protein. I'm going to, my emphasis actually, what I'm going to present here, it doesn't discriminate between any of these pathways, but in reality, we're interested in this pathway here, which is the MAP kinase pathway. But what I'm talking about actually is equally relevant for all of these, because my interest is in understanding how different effectors bind to the protein, how the lipids actually change the conformation on the proteins on the surface of the lipid, and then given that disposition, what can bind, what cannot bind, and what can be inhibited. So here is a cartoon of a KRAS on a lipid bilayer. Here I just show signal input, signal output, because it's a signaling protein. And then this section in here is it's called the hypervariable domain. And this hypervariable domain, I'm going to show you, is very relevant for the interaction. So KRAS, this is a little bit of a cartoon of how it's activated. So you have RAS, you have a GDP or GTP effector factor. This is also called SOS in many instances. So SOS actually extracts GDP from RAS. Spontaneously, GTP will bind because it has very high affinity and there is enough concentration in the cell. When it gets into the GT form, it's activated. Then, when it's activated, it's going to bind, in the MAP kinase case, to RAF. In the case of RAF, actually dimerizes, and RAF is a kinase that will self-phosphorylate, but it will phosphorylate the partner, not itself. So this is where you activate, and then a lot of things happen here. And then when you want to turn off the switch, there is a protein called GAP, and GAP very much hydrolyzes GTP, and it converts it back to the GDP form. So we are interested in understanding how this binds, how this binds, how this binds, and how all that is related to the interaction of RAS with the lipid bilayer. Here is an example of RAS in the GTP form. So when RAS is in the GTP form, this loop in here, which is called switch 1, there are two switches. The switch means that when you look at the form of the tertiary structure and the secondary structure, you actually have to, you don't follow a single chain, you have to switch. And these are typically loops. This switch gets structured or rigidified, and then it can recognize RAF. This is the RAS binding domain. So it's a small domain. Actually, RAF is a huge protein that has a kinase segment. It has 140, 180 amino acid disordered domain, and it dimerizes. So it's much more complex than what I'm showing you here. So what do we care about the positioning of RAS? Here is a cartoon that very easily shows it. So if the binding region, if the RAF binding domain binds to RAS, actually binds here, if this region is exposed, RAF can actually bind. If this region is pointing downwards, then RAF cannot bind because the membrane is there. This is slightly different from the typical paradigm where the protein is in solution and anything that is going to bind can bind from anywhere else. So there is experimental data, evidence from NMR on KRAS using lipid nanodiscs. And here is an example of two different states of the RAS on a membrane from experiment, from NMR. And here is one that the switch to, switch one in here is actually exposed, and then RAF could bind. RAF is not in this experiment. And this one in here, the switch is pointing toward the membrane, so the RAF cannot bind. You can actually have small molecules. This is a funny one because it's called compound 2. Don't ask me what compound 1 is. And compound 2 very much binds to RAS in this region over here, and this looks to some degree like cholesterol. All I want to say is that actually it has preference for the lipid bilayer, and then when it binds, it puts RAS into this conformation that is occluded from interacting with RAF. So here is a way in which you can actually design drugs, not by necessarily targeting the drug, the protein, but targeting the protein interaction with the lipid bilayer. So here is an outline of what I want to show you. I'll be going fast at some point because it's only 20 minutes. And so I want to first show what the localization of RAS is on the lipid bilayer, how this localization depends on lipid interactions, and then I'm going to show you precise maps of the RAS orientation on the surface of the bilayer and how these orientations are going to affect the interaction with effector binding proteins, and that will be all. So what I'm using is molecular dynamic simulations. These systems, whenever you have systems with lipid bilayers, and much more when you have lipid bilayers that have mixtures, the system relaxes very slowly. So actually, we have to do really extensive simulations on the system. So what I'm going to show you here is about 1.5 milliseconds of simulation. Everything has been repeated almost 100 times, so it's multiple, almost 105 microsecond simulations. There is reason for doing that based on the statistics of the system. I'm not going to go into those details. So these are straightforward MD at this level. So it's one RAS, 400 lipids. It's about 200,000 atoms per system in a 150 millimolar solution. So let me show you some of the results that we have. So here is, the first one is to describe you what is the HBR and what's so particular about the HBR of 4 RAS KB. This is the hypervariable domain. So here is, as I showed you before, the globular domain, the hypervariable domain, and here is the sequence of the hypervariable domain. So here I show you a peptide of 19 amino acids. Out of the 19 amino acids, there are 11 lysines. The peptide is actually farnesylated, so there is a lipid chain that is attached to the peptide, and that gets inserted into the lipid bilayer. And then there are other features that are also relevant. This is farnesylation. This is the 11 positive charged amino acids on the lipid, on the peptide. And then there is a methionine that actually gets inserted into the lipid bilayer also. And there are two possible phosphorylation sites, this one and this one. This one has been shown that it gets phosphorylated, so we actually also study what the effect of phosphorylation is in positioning of this fragment now on the surface of the lipid bilayer. So I'm going to show you that the disposition actually depends very strongly on lipid composition. So we're trying here two lipid compositions. One of them is pure PC. This is a sweeter ionic lipid, neutral overall. And another one that is 70% POPC and 30% POPS. So POPS is negatively charged, -1 charge per lipid. And here you see for the system when it's interacting with a negatively charged bilayer, that the system gets very close to the surface of the bilayer. So here is essentially the surface where the phosphorus, the head groups are. And this is the distance of the average distance of the C-alpha atoms in the peptide from that surface. And notice that this is really close. This is in nanometer. So this is about it's about a nanometer away. And then everything essentially gets positioned on it. When you have a lipid that doesn't have the anionic lipids, a bilayer doesn't have the anionic lipids, you see that the system gets detached. And that detachment or lack of a strong interaction with the bilayer actually shows up when you are looking at the whole system with a globular domain on it. More interestingly, if you phosphorylate, it makes no difference. So if you phosphorylate in presence of the charged lipid, the positioning very much statistically is identical. And then this peptide actually recruits PS lipids from the solution on the lipid bilayer. So we're doing a calculation where we put 30% PS, 70% PC. When we look at the neighborhood of the peptide of the HBr, we see that at some points up to 70% of the lipids are PS. So essentially the peptide is recruiting PS lipids into the neighborhood. And I'm not going to show those results, but that concentration of negative lipids actually attracts other HBRs. So the HBRs dimerizes and makes tetramers when put in a PC PS solution. Now, that what you see in the HBr, you actually are going to see that shows up when you have the whole system. Now you have the HBr plus the G domain. In terms of the plot that I showed you with the C-alpha positioning, they're almost identical when you have the G domain and when you do not. But here is the distance of the globular domain from the surface of the bilayer, actually from the center of the bilayer, for PC lipids, and then for PCPS. More interestingly, when you have either GTP or GDP, makes no difference. So if you have GTP bound or GDP bound, as far as interacting with the membranes, it makes no difference. Essentially, the distributions are identical. In looking at the recruitment or interaction with negative lipids, we have done coarse grain calculations. This data is preliminary. This is being analyzed because it's a large production of simulations. This is the calculation of 20,000 Martini simulations. So this is 20 milliseconds of simulation on systems with 2 RAS and a mixture of 8 different lipids. And what we see is that when we trace how the system behaves when it has, as a function of the amount of PIP lipids that you put in the system, around 0.5%, actually you see that as you add PIP, PIP binds to the KRAS, and about for every 2 pips that you put in the solution, 1 will bind to KRAS. And then the yellow-green line that I can hardly see, sorry, the green line is when the proteins are bound. Bound here means a swarm, is that Tajib's suggestion. And then when they are unbound, essentially there is less binding. So there is a binding of about 1 pip difference. So this is preferential binding. So it's binding above what should have been bound if it is purely random. So what this suggests is that the binding is induced by PIP, because otherwise there wouldn't have been a difference here in the binding. So let me show you the maps of the disposition of the RAS on the surface of the bilayer. I'll try to guide you through the map and what does it mean structurally. So in order to map the predisposition, imagine you put a sphere on a surface. If there is no interaction, it should be all over. If I do an angular map of what are the positions, it should be all over. So what we're doing is essentially, it's like a spherical coordinates. So we look at the tilt relative to this average position or reference position of a helix in the RAS. It's called helix 5. We use helix 2 as the second axis, and then we calculate angles relative to this. So here is a twist, and this is the azimuthal is rotating about here, and then we display that for the different systems. So here is PCPS, the 2 lipids, in GDP, PCPS in GTP, pure PC. So you see that these maps are very different. So when you have PS, when you do not have PS, you see 3 basins, but they're very shallow, and the system is really occupying a lot of the space. Still, there is some preference, and there is a basin here that you don't see in either one of these 2, but there is a basin here that is different when you have PC than when you have PS. And then these 2 maps, they're identical. So if we do a Kullback-Leibler analysis of these 2 distributions, we cannot tell the difference. So if I confuse the calculation from one system to another and I mix them up, I get the same distribution. So essentially, if they're different, I cannot tell. So from this we conclude that the GTP versus GDP, even though it changes the charge and a little bit of the system may actually be exposed, it doesn't change the preference. And in part is that the G domain is attached to the HBR, so it doesn't have complete freedom of what to do and where to go. So structurally, there are 3 basins that we can identify when we have PCPS, and there's that basin that we do not see here. These 2 basins are similar to that one, and this fuzzy one is similar to that one. So I'm going to describe you how these 3 basins, what are they structurally, and then what is this one structurally. So if we look at the secondary structure of the KRAS, we can actually, in this case, we can label label the state based on what's binding, what's interacting with the lipid bilayer. And there is a beta 1, 2, and 3 at the beginning of the sequence that actually interacts with the lipid bilayer. That's this basin. This basin in here is actually called the occluded because RAF and many other effectors cannot bind when it's in this state. I will show you that. This other basin, alpha helix 4 and 5. So alpha helix 4 is the yellow one here, 5 is the red one here. So this surface actually binds with the lipid bilayer, and the switch one, which is what you need to bind, it actually is exposed, and the system, the effectors can actually bind to it. And then alpha 3 and 4, which is the green and the yellow, also allows the effectors or RAF or something to bind. So these two are exposed. This conformation is occluded. And this is a very interesting conformation. So this is for the system when you only have neutral lipids. And when you only have neutral lipids, a region called switch 2 gets inserted into the lipid bilayer. And that interaction, actually, you can see that there are three arginines, sorry, that are involved in this interaction with the lipid bilayer. The lipid bilayer is neutral, so it's sweeter ionic. And then this region, part of the RAS, gets inserted. This conformation has never been described. So it's up to some NMR or some experiment to actually show that it actually exists. Now, there is a lot of NMR data on nanodiscs, so they're not strictly lipid bilayers, but are systems you can actually do NMR with because of their NMR experiments, because of their size. And the basins that we identify are this one, 1, 2, and 3, and their experiments that identify these conformations and these three set of conformations. Some of those are NMR. Two of them are previous simulations by Garcia. So he actually described this basin and that basin, never found this basin. And then there is experiments, NMR, and neutron and X-ray scattering data. And this is the interactions with the lipids. So who interacts with lipids and which amino acid is interacting on the different conformations. So for the alpha 4, 5 conformation, which is this one in here, you see that towards the end, there are a couple arginines and a lysine that actually interact with the lipid bilayer. For the alpha 3, 4, which is this one, in here again, lysines and arginines are interacting with the lipid bilayer. So the charges on the surface actually matters on which conformation you actually adopt in the system. And then the 1 to 3, which is this one in here, it interacts mostly at the end terminus of the globular domain. And this is the one that actually gets inserted. There's some mutations nearby this region in here that actually are also pathogenic, and we haven't made the connection between those and what the function of it will be. So now that we have these maps, I would like to see how the different effectors actually interact and what is occluded and what's allowed in terms of those effectors. So instead of doing a simulation where I have a whole new effector, what I'm doing is I'm taking the conformations of the simulation without the effectors and then see, is this conformation occluded or is it exposed for the effector to bind? So here is a cartoon. It's a composition. This is one of the simulations. This is 5 microseconds. It's actually not that much, and not much happened. This is PI3K, the kinase. This is, we take the conformation from our simulations, and we put the PI3K in the conformation that it is in a co-crystal, and then we see what the interactions of PI3K with the lipid bilayer is. When it is allowed, it's colored in yellow, and when it's not allowed, it's going to be colored in red. So here you have some amino acids insertion, and you're going to see that actually gets in. So all these are allowed conformations. So these are the system going through the 3 different basins, and then there is the occluded basin that definitely cannot happen. So it's a yes or no. Can this, if the interaction is occluded, it will be occluded because you cannot have all this domain going into the lipid bilayer. But when it is allowed, doesn't mean it's allowed. It could actually be occluded because there are other components in the system. And then we can actually analyze all the different effectors. So here is RAF. So this basing in here that is called occluded is actually occluded. So RAF cannot bind if the system is in this conformation, but it actually can be bound if it is in either one of these two conformations. It cannot be bound if it is in the conformation that we found for the pure PC lipid. And how does that compare to experiments? So here it is. These three conformations have actually been found. Simulations have found these two. And that one, there is a lot less data, but there is one critical data set that shows that. Now, this is in comparison of this occluded state. So for example, the system is occluded here, but it's not occluded there. So a 30-degree rotation, which is relatively small, can drive the system from occluded to accessible. And this is a structure from Carla Matos at Northeastern, I believe. And this is a combination of X-rays, neutrons, and even NMR data that shows what the RAF domain with the RAS-RAF domain is. I'm not barking. RAS-RAF is, and actually in that interaction, there is a little bit of penetration of the RAS binding domain into the lipid bilayer, and these are cationic side chains that actually interact with the lipid. And this is the set. Actually, we have 12 of them. So here is the map that we get. Here is SOS. This is the protein that actually exchanged the GDP for GTP. So in order to do that exchange, you have to be in this basing and partially occluded if it is in this basing. This is GAP. This is the effector that hydrolyzes the GTP. And again, you have to be in this basing because these two basins are partially occluded. And then this is RAF, which I just showed. This is PI3K that you saw in the movie. So this occluded conformation, and a lot more of it is actually occluded. It has to be in these two conformations. And this is an effector that actually recruits RAS, and it also binds strongly to RAS. So let me conclude here. So what I have shown is that the lipids defines what is the membrane adhesion to RAS. Is not the GTP versus GDP. And then it's the lipids that defines the RAS orientation. And depending on the lipids, it orients in one conformation, in one set of conformations or another. And it also has definite basings for the three conformations in the case of PC, while it has much fuzzier and shallow basins for the case of PC. And then there are orientations that occlude the binding. So you can imagine that if you favor one conformation over another, you can actually drive the system to bind one of the effectors or not. So the work was largely done, the simulations were done by Chris Neal. This work is funded by the Department of Energy in a collaboration with the NCI and also by the Center for Nonlinear Studies at Los Alamos. Thank you.
Questions?
I'm just curious, Angela, about the sauce protein. Does it phosphorylate the GDP, or does it really take it out and bring with it a GTP, or do you have to have that? Takes it out?
It has taken it out, and then
Uh-huh.
there is enough GTP, and it has a much stronger affinity than GTP. But you cannot do there is an activation barrier to exchange the so enzyme actually takes it out. It
Okay.
doesn't phosphorylate.
But then you have to have GDP in the solution to then,
GTP.
yeah.
Yes.
Okay,
Yes.
just want to be sure.
Yes. So actually the cell may decide if it activates or not because there is no
Yes,
GDP.
exactly.
And source is also activated, so it's not. Joseph.
Go ahead.
Thank you for the presentation. What do you think is the position of KRAS changed if the KRAS is mutated or BRAF is
If the
also
KRAS
mutated?
is mutated?
Is mutated.
Yes, yes.
Is this configuration will be changed?
Yes. Let me show you. We have not done this calculation, but I have to just infer from what I see in the interactions with what's interacting with lipids. So, for example, look at this conformation. Well, actually, let me look at this one. So this one, it has 2 arginines and a third arginine in here that are the key interactions with the charged lipids. So if I make a mutation and I eliminate that key interaction, it may actually not have as much propensity to be there. It may go to another side. And that's true for almost any one of the different sides. So there's some mutation, I believe, is at 155 that a lysine is mutated for an asparagine, and that's a mutant that is related also, that is also relevant for cancer. Yeah.
Will be changed.
It
It's
will,
logical.
it will change. It
Yes,
will.
yes.
It will.
It's logical to
give
expect
to the first order.
change all configuration.
Yeah, to first order, it will give preference. So, for example, if it is this one, and I eliminate the interactions that stabilize this one, then I will only have these 2, or I will have much lower population of that. That's to first order, right? We have not done this calculation because it's not all 2 well interactions. It could be more complicated than that. Yeah.
Thank you.
Thanks.
Last question.
Is phosphorylation completely steric control? That's what you're telling me?
Phosphorylation
It's
is.
going to be completely steric control? It's just the site's covered, and as soon as it's open, then you can phosphorylate, or is that...
Well, what is phosphorylated is RAF, right? So RAS, the serine at the
But
HDR
doesn't one
is-
phosphorylate the other?
Yeah. So RAF, there is BRAF and CRAF. So the hypothesis is you actually have to have either a dimer, or when RAS binds RAF, it dimerizes, and then one RAF will phosphorylate the other RAF at multiple sites, and that could actually be part of the regulation and activation for yet another kinase later on. RAF has already autophosphorylate to go from the active to the from inactive to an active conformation. In the active conformation of RAF can bind RAS. The inactive cannot bind RAS.
Right, but here when you get... But are you getting active when you get covered? You have to get exposed, don't you? You have to bind the GP.
When I assume that RAF is binding RAS, it has to be active because the binding domain is actually occluded by it's binding to the catalytic domain, and then there is this disorder domain that nobody knows what it is, and it's not a small disorder domain. It's 180 amino acids or so. So the hypothesis is this might be making multimers, not necessarily dimers, but at least dimers, and this very large disorder domain may actually be forming some conformations. So there is no crystal structure for that. People cannot express RAF and purify. They can express it, but it comes with 20 other proteins, and you cannot separate them without precipitating. So they're not crystals. Things are changing, and cryo-EM, people are trying to use that even to physically remove other proteins.
And here when you were talking about mutants, you expect the difference to be a big one or just a small one? Just
I can imagine, for example,
but imagine you just
here-
rotate a little bit to change a lot, right?
If I change these 3 or any one of these amino acids for negatively charged, imagine I change that amino acid for a serine or a tyrosine and it gets phosphorylated. Forget it. It's not going to bind the anionic lipid. So I can completely obliterate that basin. I'm speculating here. because we haven't explored any of these calculations.
Well, let's thank LANL and the 6 other speakers. We'll be back at 1 o'clock.